Hello everyone, today I am honored to introduce to you native synthetic microbial communities enhance zha-chili by boosting the fermentation capacity of indigenous microorganisms. Zha-chili is a traditional fermented food. Fermented food is an important part of the global dietary structure, accounting for 1/3 of global food intake. Traditional fermentation often relies on natural fermentation, which results in unstable product quality and great safety risks. Constructing synthetic microbial communities suitable for fermented foods is an effective strategy to solve these problems. In this paper, a synthetic microbial community consisting of 2 bacteria and 3 fungi was designed and constructed using zha-chili, a traditional fermented food, as a research model. This study has 3 highlights. 1. A native synthetic microbial community was constructed for zha-chili, which can improve the flavor content and shorten the fermentation time of zha-chili. 2. The synthetic microbial community significantly changed the composition and succession pattern of fungal community in the fermentation system of zha-chili. 3. The synthetic microbial community may enhance the quality of zha-chili by increasing the contribution of other native microorganisms in the community to the flavor substances. Firstly, we need to construct a synthetic microbial community from zha-chili. Statistically significant synthetic microbial communities were identified through preliminary studies, and the intersection was taken to obtain a list of core microbial communities. We screened and isolated strains from several batches of zha-chili products with different fermentation times and obtained 5 target strains. The fermentation effect of the 5 strains comprising the synthetic microbial community varied, but the trends of their physicochemical factors were similar to those of the CK group. A total of 142 volatile flavor substances were identified, and we artificially screened out more than 30 edible flavor substances with pleasant aromas. Overall, single bacterial inoculation improved the production of some volatile flavor substances, although it did not change the trend of the main physicochemical factors. The trend of physicochemical factors in the experimental group of synthetic microbial community was consistent with that in the CK group. The glucose content in the LP group was significantly higher than that in the CK group at the beginning of fermentation, implying that inoculation of synthetic microbial community could rapidly decompose starch to produce reducing sugar to promote the fermentation process. The content of specific flavor substances in the experimental group of synthetic microbial communities was generally higher than that in the CK group. At the later stage of fermentation, the LP group produced less hexanoic acid than the CK group, suggesting that the LP group produced more flavor substances while producing fewer undesirable odors. The macrogenome sequencing showed that most reads were identified as bacteria, with Escherichia coli, Bacillus immobilis, Salmonella, and Corynebacterium as the main dominant genera. The bacterial community succession in the experimental and control groups was approximate. The main dominant genera of eukaryotic microorganisms were Pichia, Saccharomyces, and Zhao. The introduction of synthetic microbial community significantly altered the fungal community succession. We observed that the RC index of the LP bacterial community in zha-chili was close to -1, suggesting that the similarity between the bacterial communities was high and mainly driven by deterministic processes. In the community succession of fungi, the RC index of the CK group was close to -1, suggesting that the fungal communities were less variable than would be expected from stochastic expectations and showed similarity. However, in the LP group, the RC index of the fungi showed a different pattern, with some sample sites close to 0, suggesting that the succession conformed to a neutral model. In this study, we draw an important inference that synthetic microbial communities may improve the overall microbial system by influencing the functions performed by other microbes in the community. Correlation analyses of the CK group showed that Saccharomyces, Pichia, and Candida showed strong positive correlations with the major flavor compounds. However, Pichia, which dominated the LP group, did not show strong correlations with the major flavor compounds. On the contrary, other fungi ranked in the top 10 in terms of relative abundance contributed more strongly to the main flavor profile positively. We interpret this phenomenon as a contribution of the inoculated synthetic microbial community. Summary. In this study, we screened and established synthetic microbial communities associated with the fermentation of zha-chili, and verified the inoculation effect of synthetic microbial communities in subsequent single. We found that the modification of zha-chili by synthetic microbial communities resulted in a reduction of fermentation time. The synthetic microbial communities altered the composition and successional pattern of microbial communities in the zha-chili fermentation system, especially the composition and successional pattern of other fungi. It was concluded that the synthetic microbial community could improve the function of the whole microbial community by regulating the role of other microbial species.